Hi everyone, in this video I'm going to go over the exam 3 practice questions that Dr. Zillman posted so you can study. This is a good way if you weren't able to come to one of our sessions to check your work, see if you understand the questions and the answer options, and hopefully you got the answers correct. So the first one, which of the following techniques could you use to study the level of gene transcription in mouse embryos? So this is testing your knowledge of the different techniques that were used. So let's go over each one. So comparative genomics, remember that is when you're analyzing the relations of a genome sequence of two or more species. So comparative, it's in the name, but comparing two or more. RT-PCR or reverse transcriptase PCR, that is when you're looking for a specific gene transcription. So you just want to look at one transcript and you're looking at a specific gene. The next one, Sanger, also known as dideoxy sequencing, that's the one with all the little colors and the little blocks and pieces. This is when you want to determine the exact sequence of a gene. You're trying to identify the specific gene that is associated with like different diseases or something like that, but you're looking at a specific sequence. This is when you're also looking at the nucleotide level. So that's when all of those different markers are colored, and then they go back and make a key and figure out that, for example, red is associated with A, and so on and so forth. Okay, and then the next one is just regular PCR. Remember that you're starting with DNA, and you want to amplify your gene of interest, or your GOI. And this is also when you are looking at how much DNA is present, so you're looking at abundance. Okay, and then the very last one is whole genome sequencing, and this is when you are looking at multiple regions or you're looking at the entire thing. Okay, so then based on this question, if you are wanting to look at the level of gene transcription in mouse embryos, you would be looking to use RT-PCR because again, you're looking at a specific gene transcription. So that is why you would use it, level of gene transcription. And that is why you would select B as your answer. So I'll include specific gene transcription as well. So yes. So B is your answer. Okay. Moving down to the next one, next generation sequencing. This is when you add your nucleotides and then you stop and you pause, you take that picture, you cleave and wash, and then you redo it. This is the type of sequencing where you take the pictures and then you look at all of the sequences in parallel. So the correct answer for this one, I'll give you a minute, pick which one you think it is, stop the video, and then resume to check your work. But the correct answer for this one is letter B as well. And I'll explain why the other ones are not true. So letter A is not correct because it says that it's using the DD and DPs, but it doesn't use it to terminate DNA synthesis. And then letter C is incorrect because it's called high throughput sequencing, not low throughput. And then D is incorrect because it says it uses the existing strand directly. It actually relies on synthesizing a complementary strand of DNA while detecting the added nucleotides in real time. And then letter E is incorrect because it says that it requires you to first generate a contig, but it actually doesn't require. It makes many small fragments independently, and then it makes the contigs after the fact. So that is why letter B is correct. Okay, the next one, base excision repair. Definitely become familiar with what base excision is. There's a couple, I think there's a video posted about this, so go watch Tim's video explaining exactly what base excision repair is. However, in base excision repair, predict the consequence of a mutation in DNA glycosylase. So remember what glycosylase or DNA glycosylase does. So this is your keyword here. And remember that it's responsible for recognizing and removing the damaged or incorrect bases by cleaving the N-glycosylase bond between the sugar and the base. So I'll just say recognize and remove the damaged or incorrect base pair. But again, review more in depth kind of what base excision repair is and DNA glycosylase's role in it. Okay, so the answer to number 4 is letter D. The nucleotide would never be excised and repair would never occur. And this is because if there was a mutation in it, the damaged base would never be able to be removed. So this couldn't create the AP site, and the repair process would never proceed. So again, if there is a mutation in glycosylase, glycosylase is the one that recognizes and removes the damage. If you're not able to do that, then the nucleotide would never be able to be excised or repaired because the thing that does it is mutated. Okay. Next question. 3 types of genes are known as cancer critical genes. Which of the following is true? You have letter A, mutations in caretaker genes are considered indirect contributors as they allow mutations to accumulate more quickly. Mutations in caretaker genes increase the rate of cell division directly, leading to cancerous growth. And then lastly, the same types of mutations in tumor suppressors, proto-oncogenes, and repair proteins can lead to cancer transitions. So pause the video, again, pick which answer you'd like, and then I will show you which one's correct. And the correct answer is letter D. Choice A and letter C are true. Caretaker genes are considered indirect contributors as they're not like directly impacting cell growth. And then letter C is correct because tumor suppressors, proto-onco, and repair proteins can all lead to cancer transitions. The reason why B is not correct is because it says that caretaker genes increase the rate of cell division directly. However, they don't directly drive cell division, they just prevent DNA damage from accumulating. So that directly is what is incorrect about letter B. Okay, last multiple choice question. Imagine you have a chromosome or a genome where chromosomes are named HIJK instead of using numerical designations. If a normal diploid genome is represented by HHIIJJKK, what would HHIIJKK represent? Okay, so remember with this question, it's testing, do you know the difference between SOMI and ploidy? One way I like to remember is thinking about Down syndrome or trisomy 21. That usually helps me figure out which one I'm dealing with. So remember that SOMI is 2n plus 1, where ploidy is 2n. So in SOMI, only one chromosome is duplicated. Whereas in the other one, ploidy, the entire genome, or all of the chromosomes, are duplicated or impacted. So again, like I said in the example of trisomy 21, SOMI, so thinking of Down syndrome, trisomy, you have only the 21st chromosome being duplicated. It has an extra chromosome where all the other chromosomes are normal. They don't have that extra chromosome. Where ploidy, it would be, so triploidy would have all of the chromosomes in the genome would have 3 copies instead of just one single chromosome being affected. So in this case, you have your normal set is diploid, so you have 2 of everything. But in this one, you have a J that is missing its other pair. So you only have 1 J instead of 2. So the answer to this one would be monosomy. Because again, only one chromosome is being affected. It would be monoploid if all of these letters here were only had one. So if it was just H, I, J, K. But because H, I, and K all have their pair or like their diploid match, so there's 2 of the letter, it makes it somy because only one of the letters is affected. Or in this case, chromosomes, because the letters are being represented by chromosomes. So again, only one chromosome is affected, just like in Down syndrome. Okay, we talked a lot about mutations this semester, which always makes me a little nervous. Why aren't we sick all the time? Describe the sources of mutations in our lives. So when you're thinking about mutations, start thinking about the different types. So that Dr. Zillman talked about. So we have some, we have both sources and why they don't matter. So I'll start with why they don't matter. Remember that sometimes they might be in a non-coding region. So if a mutation occurs in a non-coding region, then it's not going to get read, and most of the DNA is made up of non-coding regions. So it doesn't matter, because if it's not getting read, it's not getting translated, it's not turning into a protein, it doesn't have any effect. You also have a diploid advantage. And this is when you have 2 copies of something. So if you have a bad or a mutated copy, it's okay, because your second normal copy that is functional can typically like make up or supplement for the bad copy. Another reason could be the mistake could be corrected. This means that DNA polymerase, it has proofreading activity, so it can basically go back if it notices that there's a mistake. It can backtrack a little bit, fix the mistake, and then keep moving forward. And then another one is that it might be a silent mutation. This means that although the sequence of the nucleotides may be changed, it might have no effect on the gene's function. This is because the genetic code is repetitive and there are things called wobbles, so different letters or a sequence could still potentially code for the same amino acid. Therefore, there's no like drastic difference or impact on the protein itself because it's being coded for the same thing. Okay, next I'm going to talk about, and of course, review all the different types. I'm just giving a few examples of some. Now I'm going to go to the different sources of mutations. So some sources include just DNA replication errors. And this is just because our cells continuously replicate their DNA to replace old or damaged cells. And during this process, the double-stranded DNA separates, and each strand then is copied to form a new double strand. But every time the cell divides, it must copy its entire DNA sequence. And occasionally it might misread or miss a piece, which might lead to mutations. You also have mismatches from tautomerization. So mismatch. And this is just when the structure of the DNA building blocks change momentarily, and this can lead to incorrect base pairings. You also have like external factors such as like UV radiation. This can impact our cells. It induces the formation of pyrimidine-dimers, which then can distort the DNA structure. You also have things such as carcinogens. So some chemicals, analogs, they can mimic nucleotides and be mistakenly incorporated into the DNA, and this can lead to incorrect base pairing in subsequent replication. Okay, and then you also have DNA slippage. And this is just when like insertions or deletions can cause mutations. So it kind of slips in and out of the sequence, the different nucleotides. You also have things such as just temperature. So our body temperature is very high. So um, your own body heat can cause formation of abasic sites. Um, so it can cause mutations. And then lastly, oxygen or oxidative deamination. So just the presence of oxygen itself can impact cells and lead to mutations. Of course, this is not an exhaustive list, so do go over the different examples in the slides. Here's just some that I came up with. Okay, then the next question. Two types of mutations are particularly notable for their roles in leading to cancerous transitions, proto-oncogenes and tumor suppressors. What do these genes normally do? How do mutations change their functions? So remember that the difference you have like proto-oncogenes. So proto-onco is actually the regular name. It is the non-cancerous version. And these are the ones that it accelerate or are the accelerator pedal for cell growth. So normally these genes help cells grow and divide and make a new cell, or they just help it stay alive. So grow and divide, or just simply stay alive. So when there's a mutation though, that is when you get just the name onco. So no proto, just regular name onco. This is when there's a mutation, it can be activated. Basically, cell growth is activated when it's not supposed to, which is when it leads to too many copies being made. So growth is activated. When it's not supposed to be. And then this is what leads to your too many copies. And then on the other hand, you have tumor suppressor genes. And regularly, these help keep the cell from dividing too quickly. So normally, it acts as like the brake for cell growth. And it slows down cell division or tells the cell to die at the right time. So it slows down or tells it to die, or apoptosis. So a mutation in a tumor suppressor can basically lead to cell division getting out of control because there's nothing telling it to stop. Right, and then that is what the difference between proto-onco and tumor suppressors. Definitely look over your slides and your textbook to get a more in-depth definition and kind of explanation, but at a quick glance, that's what the two are. Okay. Next question is kind of similar to what I've explained in the multiple choice, but chromosome level aberrations can dramatically change the genetic content of a cell. What is the difference between an individual with trisomy? So like trisomy 21, trisomy, and an individual that is triploid. Consider the potential consequences on the cell in terms of both genome content and gene expression. So again, like we talked about earlier, trisomy is where you have an extra copy of just one chromosome. So that would be your 2n plus 1. So all the other chromosomes in the genome would be 2n, but you have one chromosome that would have that extra copy. So again, in trisomy 21, the 21st chromosome has two copies, but it has an extra copy where all the other chromosomes in the body just have 2n. So with that being said, there would be gene expression only in that chromosome that's being affected. So, whereas you have triploidy, the entire set of chromosomes is affected. So there's 3 copies of all chromosomes in your body, or 3n. So this is 3n, this is 2n plus 1. So in this scenario, triploidy, instead of having 2 copies of each chromosome, and having just like an extra copy. So here, I'll draw it over here. So this is just 2 copies. You know, all the other chromosomes, where trisomy, you have your 2 copies, which is represented by 2n, but you have this additional copy here. Where in triploidy, all of your cells have 3 copies. So again, trisomy, there would only be expression only on the chromosome that's being affected. So you'd have like for an example of trisomy 21, you'd have 3 21st chromosome being expressed. So 3 copies of the 21st chromosome being expressed. Whereas in 3n, the genes, all the genes, not just the 21st that's being affected, all your chromosomes would have 3 copies, which would lead to expression in all of the chromosomes. So again, all chromosomes, 3 copies, and all of them would be expressed. evenly. Whereas in this one, again, I'm just giving the example of trisomy 21. In trisomy 21, or just trisomy in general, you have all of your chromosomes have two copies. But that extra chromosome that you have, but that one chromosome that you have an extra copy of, would be expressed more. There would be more expression of that 21st chromosome because there's just more of them. Hopefully that helped. Again, consult your textbook if it's a little bit easier, but let's move on to the next question. Your cousin just turned 40 and is pregnant with her first child. Her doctor recommended prenatal testing due to her higher risk of chromosomal aberrations. She asks you why this is necessary for her, but not for her 25-year-old sister. Please explain. So, at birth, females have all of their oocytes, and meiosis resumes with each menstrual cycle. Therefore, all of the paired chromosomes are maintained for decades. So essentially, here's your egg, and they're all lined up kind of in the middle, just getting ready to be paired up, but they're all waiting at this point. So while they're not menstruating, while they're in between their cycles, this is what their eggs are doing. They're all just lined up, ready to go. However, because of this, the hypothesis is that non-disjunction increases with the length of time that chromosomes have to be paired, or the time that paired chromosomes have to be maintained. So there's a higher probability that the paired chromosomes will break apart over time, which will lead to disjunction. This is, so if you think about it, the longer something has to stay attached, or like the older that you are, the larger the increase in error there is. Because think about it, if these eggs right here have to be paired together, just paused for 40 years versus a sister who is only, her eggs only have to be paused for 25 years, there's a lot more time that these eggs in the 40-year-old will have time to pair up incorrectly, versus the 25-year-old sister who has less time, she's younger, less time to make mistakes. So the cousin, who's 4 years old, is older than the 25-year-old sister. So this just means that her eggs are older and have a higher chance of error occurring just because of time. Again, more time equals more likely you are to have some kind of chromosomal aberration. So again, I'll write so longer. So again, I would write a much longer explanation of this, but longer they stay attached. The more likely or the higher the probability that an error will occur. Okay, next question. We're talking about the difference in syndromes. So, individuals with Turner syndrome have an XO chromosome designation, and individuals with Down syndrome have a extra copy of chromosome 21. Each of these is designated a syndrome because of the dramatic range of phenotypes seen in individuals. So this question is asking, why do we see such a broad range of phenotypes that affect so many different systems, and why are such chromosome level changes so debilitating? So when we're looking at why do we see such a broad range of phenotypes, this is because each syndrome has a different chromosome with its own unique genes that are impacted. So for example, trisomy 21, we see an extra copy of the 21st chromosome. Therefore, the genes expressed on that chromosome are seen more than the other chromosomes, which only have 2 copies, not 3. So again, if you're looking at this, so all the rest of the chromosomes, again, only have 2 copies, where chromosome number 21 has 3. So remember, on a chromosome there's a bunch, here's my drawing of a chromosome, but on a chromosome there's a bunch of different genes that are associated with each chromosome. So for example, let's say this is the 21st chromosome. Let's hypothetically say that I don't know, I'll say like hair and I don't know. We'll say hair and eye color. Again, this is not at all accurate, but let's say that hair and eye color are on the 21st chromosome. The hair and eye color would be, those genes would be expressed more than maybe other types of genes because there's 3 copies of them versus 2 copies, which are in everything else. Again, that is not accurate. I have no clue what the 21st chromosome, what genes it has on, but that's to help you visualize. So they affect so many different systems because the chromosome that's impacted. So there's a variety of different genes on each chromosome, therefore, the genes on that chromosome are being expressed more just because of the extra copy. So again, each syndrome has different chromosomes that are affected. And those chromosomes have their own genes that are specific to those. So that's why those are impacted. So again, elaborate a little more, but again, just remember that there's a wide range because each syndrome has a different chromosome with its own unique genes that are impacted. Therefore, the genes that are expressed on that chromosome are seen much more than others that only have 2 copies. In the example of trisomy 21. Okay, and then the last question, why are chromosome level changes so debilitating? They're so debilitating because a chromosome has a lot of different genes that are impacted. So a lot of genes are impacted. And this would end up, we would see changes in expression that interrupt the balances of interacting proteins that affect important pathways like cell division. So we're also thinking about balance, like gene balance, and how if gene balance is disrupted, that will then disrupt different proteins and pathways that are important for cell division. Okay. Alrighty, and then the last question is over Punnett squares and finding the genotypes when you are given the phenotypes. This can be a little tricky just because it does take a little bit of time, but if you start memorizing the ratios, it should definitely help. So kind of start memorizing, like if you cross a homozygous recessive times a heterozygous, a homozygous dominant times a heterozygous, like start kind of looking at those patterns. So here's 3 families. So the first family, or first thing that you should do is look at what you're given. So you're looking at the different types of tails, either short stubby tails or long tails. So in this family, you have only stubby tails, and you have none with long tails. So I always make a note of what I see. So I'd say stubby tail only, or you have a 1-to-0 ratio of stubby to long. Okay, and then in family 2, it looks like you have a pretty even split, or a 1-to-1 ratio of stubby to long. Here and here. And then in family 3, looks like you have a 3-to-1 ratio. 3 being the long and 1 being the stubby. So you can either say 3-to-1 or 1-to-3, doesn't really matter. I'll put 3-to-1 for this. But long and then stubby. Okay, so then once you have those, you kind of need to figure out which one is dominant, which one's recessive. So here you only have stubby tails. So if you're looking at which would be which, it looks like you would have, again, all stubby tails. So this individual, we would say that stubby, based on the families, we would say that stubby is recessive and long is dominant. So I'm just using T. You can use whatever letter you like, though. But because based on the ratios of the families, stubby would be recessive and long would be dominant. So in order to get a family with all stubby tails, again, all recessive traits, you'd have to have individuals, 2 homozygous recessive individuals, because that's what would get you a completely recessive or stubby-tailed family. Any form of any big T or long would produce a long-tailed individual. So this is what is going to give you that stubby only. Okay, and then in the next family, it looks like you have a 1-to-1 ratio. So again, thinking back to kind of memorizing those ratios, it's important to know that if you take a heterozygous individual and a homozygous recessive, that is what will get you that 1-to-1. So half of them would be heterozygous and the other half would be homozygous recessive. So again, you would only have, so in this top case, you'd have 100% little t, and then in this bottom, you would have half of them would be big T, little t, and half of them would be homozygous recessive little t. You can also use percents or ratios, whichever you would like. Okay, and then lastly, we're looking at family number 3, and we see that there is a 3-to-1 ratio. And recall that if you cross a heterozygous individual times a heterozygous individual, that is what is going to give you that 3-to-1 ratio. So it's going to give you that 3-to-1 phenotype. So physically, they're going to look 3/4 are going to look the same, 1/4 will look different, but it's going to give you that 1-to-2-to-1 genotype. So again, genotype, you'd have 1 that's homozygous dominant, 2 that are heterozygous, and 1 that's homozygous recessive. So that's where you get your 1-to-2-to-1. However, your 3-to-1 comes from these 3 right here. So your homozygous dominant and your heterozygous individuals would appear the same because they all have that dominant or capital T long-tailed trait. So that is how they would physically look, all the same, giving you that 3. So again, just kind of start looking at your ratios, start memorizing kind of what different outcomes, like what crossing certain things will give you, and that should help as well. So for, again, family number 1, you would be crossing a little t homozygous recessive times a homozygous recessive. That would be your first family. Your second family, you would be crossing a heterozygous individual times a homozygous recessive. And then your third family, you would, your third family, you would be crossing a heterozygous individual times a heterozygous individual. Okay, so that's figuring out the parents. And then it says in the lab, you develop populations that are true breeding for long-tailed trait and the stubby-tailed trait, which are presumed homozygous. If you cross an individual from each tank, what would the genomes and phenotypes be for the first generation of offspring? So they're saying that you are crossing that are presumed to be homozygous for each trait. So, right, number 2 right here. So that means you would cross a stubby tail, which would be right here, or excuse me, a stubby tail, which would be right here, and then your long-tailed trait would be represented by the 2 capital T's because they're both homozygous or the same allele, same letter. So if you crossed a homozygous dominant times a homozygous recessive, this is when you would get all heterozygous individuals. So big T, little t across the board. So these individuals would all be long-tailed because they have that dominant trait. Okay, and then lastly, number 3, if you let individuals from the first generation mate with each other, what would be the genotypes and phenotypes represented in the next generation of offspring? So they're basically saying in this first generation right here that you just created, so these individuals, if you crossed a heterozygous individual times a heterozygous individual, what would you get? And that is when you would see that 1 to 2 to 1 or that 3 to 1 ratio. So again, you'd see the genotype would be 1 to 2 to 1. You'd have one that's homozygous dominant, 2 that are heterozygous, and then 1 that is homozygous recessive. So you could say 1/4, 1/2, or 2/4, whichever you would prefer to write, and then 1/4 here. However, phenotypically, you would see that 3 to 1 ratio because 3/4 would be long-tailed because you have these 3 here that would be represented by the long tail because they have the dominant T or the dominant trait of having long tails. And then 1/4 would be short-tailed. Okay. All right. I hope that made sense. I hope you kind of figured out what the different ratios are and what is long and what is short. I know this can be a little bit difficult, but start to memorize the different ratios. The more you can start looking and memorizing and recognizing the different ratios and what they produce, and what like the parents would be, it really, really helps. So it's really about practicing and getting familiar with the different ratios and homozygous versus heterozygous, recessive versus dominant, what crosses with what produces like what. So really focusing on the outcomes and the different parents. So again, I hope this video helped. If you have any questions, of course, reach out. But good luck on your test.